Welcome everyone to our webinar on calculating polygenic scores with the Polygenic Score Catalog Calculator. So I'm Sam Lambert. I'm an assistant professor at the University of Cambridge, and I'm also a visiting researcher down the road here in the UK at EMBL-EBI, which is kindly hosting the webinar today. So in today's webinar, you'll hear from me, the Polygenic Score Catalogs has the two institutions, Cambridge as well as EMBL-EBI. And you'll also hear from Ben Wingfield, who's a genomic data scientist and scientific software developer who we work on this collaborative project with. So the work today isn't just the work of us, it's the work of a wider PGS catalog team spread across lots of different institutions and lots of different people. And it's just I like to start it off by thanking all the people who contributed to the project before we get going with seeing what we did. So just to bring people onto the same page about polygenic scores, a polygenic score is basically a single value that quantifies an individual's genetic predisposition to a trait or disease of interest. And you might hear them called polygenic risk scores, genetic risk scores, genomic risk scores, genome-wide scores. But they're all different flavors of the same thing. Usually the most common is polygenic score or polygenic risk score. And people usually use polygenic risk score to sort of say that it's predicting a disease or clinically relevant trait. So it's your risk for that. So basically, there's lots of different methods out there that exist to select the variants and weights that are used to calculate an individual's predisposition. And once you have those weights, I'll just go over the general polygenic scoring approach. So I'm going to start with a sort of toy example from some GWAS data. And so basically what would have happened is someone gets a polygenic score, which is the list of variants that are significantly associated with the trait you're trying to predict. You have the variant information, so it's RSID or chromosome position, the alleles, which allele causes an increase or decrease in risk for the trait, and then the effect sizes. Usually it's like an odds ratio if it's a disease, but then in an additive linear space, it's the log odds ratio. So here you receive an existing polygenic score for a trait for these variants and these effect sizes, and then we wish to calculate the polygenic score on different people. So here's an example of someone in a tuxedo. They have these genotypes, their individual level genotypes, and then if we count the risk increasing dosage, or the effect allele dosage, we can see they have one copy of most of the risk alleles and then two copies of some of them. Then if you multiply the risk dosage by the effect size, you get the per variant contribution and you sum it up and that gives you a polygenic score for this person or this individual. So this person might have sort of a lower polygenic score. You can see that the theoretical maximum, if you had all the risk alleles, your polygenic score would be 2.62. So this person, maybe they have like a medium or average polygenic score. And this person on the right, then you can see they don't carry many of the risk alleles. So they would have a lower polygenic score. Theirs is 0.32, so very close to the minimum polygenic score you could achieve in this scenario, which is 0. So basically, the variants you use then allow you to calculate a person's polygenic score, the number that corresponds to their genetic predisposition to a trait. But the number is sort of scaleless and isn't so interpretable. So what you need to do is to look at polygenic scores in a population of people. So if we calculate a polygenic score for a bunch of different people who are of the same genetic ancestry, what you would see is that the polygenic score forms a normal distribution in the population. So that is, there's lots of people who, just because of how like assortment works of alleles and also just selection, you basically have lots of people in the middle who have sort of an average polygenic score, and then there's people in the tails who might have lower or higher than average polygenic scores. But for an individual, we wouldn't know based on that number where they sit in the distribution. You basically have to compare individuals to a distribution to say what their relative polygenic score is or their relative measure of genetic risk. The splitting of the distribution is somewhat arbitrary as there's no natural cut points in a normal distribution like this. But risk can often be expressed as a percentile or a comparison to average to the mean polygenic score in a population. But this only allows you to calculate a polygenic score, but a polygenic score then needs to be validated to be predictive of its target trait. So you have to establish the predictive ability of the polygenic score in external populations and on the phenotype you wish to predict. So early examples, this is one of the earliest polygenic score papers in humans, where they take a BMI, a GRS for BMI, and then they stratify the individuals into different buckets. And you could see that the mean BMI increases with the mean number of weighted risk alleles, and you could use that to predict obesity. But more recently, people are interested in sort of longitudinal phenotypes, time to event phenotypes, and there you could basically split the distribution up into individuals with lower, medium, or higher than average polygenic distribution, and see that these people have different trajectories of risk over the life course. So this is an example of coronary heart disease polygenic score and the follow-up in individuals from midlife to late life. And you can see people in the highest risk bucket have the most CAD events over time. So you need to take these polygenic scores and then evaluate them. But a lot of the information about what polygenic scores is quite variable. Often there's multiple different scores for the same trait, so they're developed using overlapping data sets, but the metrics aren't directly comparable between scores and studies, oftentimes because even if they use the same starting data, they might split it up slightly differently and test on different subsets of the population. And we know that polygenic scores have some sort of reduced accuracy in populations that are dissimilar to the initial cohorts. So if the GWAS or training data for your polygenic score has only European ancestry people, you would expect reduced performance in other ancestry groups just because they're dissimilar in some way by allele frequencies and LD mainly. And before we set out on our work on the polygenic score catalog, actually a lot of the variant information needed to reapply polygenic scores to new samples was often not shared. So this brings us to why we created the Polygenic Score Catalog, because the open sharing of polygenic scores goes beyond good reporting to enable the downstream reuse and comparison of existing polygenic scores. And this has been really helpful for resources and communities like the GWAS community, and the GWAS catalog has been a real instigator of lots of downstream research and open science. So the Polygenic Score Catalog, we developed it to have an open database of polygenic scores that can be calculated on new samples, along with relevant metadata that you need to apply and evaluate them in new samples. So the website is pgscatalog.org. And here's a slightly out of date slide, actually, I should have said, but there's actually over 5,000 different polygenic scores for over 650 traits right now. And it's developed with our colleagues at EBI at the GWAS catalog. So within the Polygenic Score Catalog, we collect lots of different information that's needed to understand which polygenic scores can then be used in new data sets and applied to new samples. So for each polygenic score, we capture information about the trait it predicts, how the polygenic score was developed, so which computational methods were used, as well as the samples used to develop the polygenic score. So which GWASs were used, and we link that to the GWAS catalog study IDs, and which samples were used to develop the score and fine tune it. So that is to select the variants and weights. And we describe these consistently in terms of their sample size, the ancestry, age, sex, and follow-up time, and which cohorts were used to avoid overfitting and overtraining polygenic scores. We then provide a polygenic score scoring file, which has all the variant information needed to calculate the score on new samples. And this is basically a minimal specification of the variant and where it is in the genome along with the genome build, as long as the effect allele and the effect weight. And then we also do extra work to basically make sure that the polygenic score that we provide can be calculated in build 37 or 38 by making sure that all the variant information, so position, chromosome, alleles, are in the file to make variant matching easy when you go to match the variants in the polygenic scores to the variants in your samples. So that provides the score, and then we provide information about how well the score performs in evaluation cohorts, and we curate this from the original paper as well as subsequent evaluation papers. And this includes information about the trait and what covariates they might have used in the polygenic score evaluation. You can access all this data on our website, pgscatalog.org. You can browse the scores, publications, and traits. Here's an example of how you can browse the diversity of traits in the PGS catalog. And then the data in the catalog is also accessed through bulk downloads, a REST API that can be used to programmatically access and query the catalog, as well as just on the FTP, all the data is there that you can just download direct and reprocess it yourself. So the data in the PGS catalog, it enters this sort of fair data journey where the data is findable, accessible, interoperable, and reusable, where we find polygenic scores in the literature, and we might ask authors to submit their data to us. But authors also submit data direct to us so that we can make it, validate it, upload it into the catalog, and release it so that it's accessible for calculation through the calculator tool, which we're going to go over today. And we also provide the scoring files in a harmonized file so that they can be used in other tools. So data in the catalog is used on the Michigan imputation server by other tools like PRS Calc and GenoPred. So the data in the catalog allows you to answer the question about which score should I calculate, because basically there's lots of different scores for each trait. So I'm a cardiovascular disease researcher, and usually I'm focused on polygenic scores for coronary artery disease or hypertension, and there might be multiple different scores that meet that criteria. But basically, on each page, the score has information. You can browse all the information about who the score was developed in, what GWASs were used, what performance metrics, and basically the rich metadata allows you to determine if a particular score might be fit for purpose and calculating on your own samples. So this is just a scroll through, but any PGS catalog score page will have information about this. One thing is that, as I mentioned before, there are limitations to the usability of polygenic scores, and it is important to note these and interpret polygenic score data with caution. Most genetic data and existing GWAS studies are from predominantly European ancestry participants. And so what happens is that if you build polygenic scores exclusively from these data, it results in reduced performance in non-European ancestry groups. And this has been shown in many papers, but I think it's clearest in this Martin et al. Paper from AGHG and Nature Genetics. But what you can see is that the score will have reduced, but not ablated predictive value in non-European ancestries if it composes only European ancestry training data. And it's important to note this and to maybe seek scores that have included data from your ancestry group that you wish to apply the score to, so that you can find a score that's more applicable to the population in which you wish to predict genetic predisposition. So we provide some tools to do this on our website. You can browse the ancestry information. We provide a visual summary of the ancestry makeup of the GWAS samples, as well as the evaluation samples. And you can filter this. So here you can take in this associated polygenic scores box, you can filter this to find scores that have been previously evaluated. In this case, I've substituted to be previously evaluated in African ancestry participants, so that these scores might have performance metrics in African ancestry groups, so that you know that the polygenic score could be applied to those there. Another, so this allows you to find the scores, but once you calculate the scores, there are some problems that PGS distributions are slightly confounded by genetic ancestry. So as I mentioned before, when we score a polygenic score in a bunch of people, it will be normally distributed. But our goal is to accurately determine an individual's relative risk for a disease or trait X using a polygenic score. So when you do this in a diverse cohort of individuals and that are of different ancestry groups, what you can see is that if we plot the PGS distributions in people of different self-reported ethnicities or predetermined ancestry groups, there often is this shift in means and different standard deviations in these different population groups. And this is because changes in allele frequencies and LD patterns across populations cause shifts in the PGS distributions. And we need methods to basically, and if we just use the raw PGS, which is on the x-axis of this graph, we would get a biased result for certain populations. And we would say maybe the European population, which is at the far this left, has the least genetic risk for this disease, but the orange population at the top, which is African, has the highest. But these means and shifts don't actually correspond to differences in the trait values or prevalences of a disease like type 2 diabetes in the populations. It's purely due, well, to some extent, it's due to genetic ancestry. And this is a barrier to the accurate interpretation of PGS. So there are methods that exist to decorrelate polygenic scores from these differences in genetic ancestry to give each individual a calibrated relative risk for disease X using the polygenic score. And so these come in two flavors, and I'll go over them as these methods are implemented in the software you'll hear about later in the webinar. So there's first method is sort of an empirical method where we take this person and who's pink, and we compare them to a population reference panel where we've scored the polygenic score. And then because this person is pink, we compare them to the pink population to see within this distribution of polygenic score in pink ancestry people, does this person have a higher or lower than average polygenic score, or what percentile of this pink distribution do they sit in? And this is implemented in this is similar to how you analyze a cohort of genetically similar people, but on the individual level, it was originally implemented in ImputMe, which is a software with the reference there. But this method requires assigning a person a most similar population, either by ethnicity or by using PCA projection and clustering. And it basically only compares them to one population. So this is likely to fail if the person is admixed or doesn't sit perfectly if the population that's they're part of is not perfectly in the reference panel of individuals we're comparing to. So a second method of polygenic methods to sort of normalize polygenic scores with genetic ancestry information uses continuous information about a person's reference, a person's ancestry. So here you can see, like in the graph on the last slide, the polygenic score that we're considering is stratified. It has different means and standard deviations in the different populations, but we can correct for this by basically regressing out the differences about where someone sits in PC space and their expected polygenic score. And we can do this to make sure that the means of the polygenic scores overlap between different populations. So this is what we call regressing out the first moment of the distribution. And this was in the Kara et al. Paper in 2019 originally. And then we can do a second and what you can see in that first graph is basically the distributions have the same mean, but they have different spread, different variance. And we can also make sure that the variance is the same in different populations. And this is by regressing the residuals of the first regression to make sure that each population has the same mean and standard deviation for their polygenic score, so that it's a perfect measure of genetic predisposition, relative and irrespective of your genetic ancestry. And this works by then taking the pink person, seeing where they sit in the PCA space, calculating their expected polygenic score, and then returning the Z-score that's based on regressing at the mean or variance differences that we would expect based on this person's genetic ancestry. And these methods have actually been used to report polygenic risk score and polygenic score results in different trials. So the PGS adjustments make these relative risk estimates more consistent across ancestry groups. And this is on the right, you can see a link to our documentation that provides a longer written summary of these methods and why they're implemented in our calculator software. But basically, it converts this raw PGS sum to a relative genetic predisposition of an individual irrespective of their genetic ancestry. But it is important to note that the adjustments don't solve the transferability or performance differences between ancestry groups. It only makes the estimate of their predisposition more accurate, but not the effect size of what that means in terms of which diseases they're more likely to get or which or their predisposition for higher trait values. So with that, I've given you a bit of an introduction into what sorts of data we have in the PGS catalog and what methods exist to calculate PGS and what we need to watch out for where we wish to calculate PGS. And so in the next section, my colleague Ben is going to be describing how to calculate polygenic scores with our PGS calculator software, which automates all these steps and makes it accessible and reproducible. So with that, I'll turn it over to Ben.
All right, thank you, Sam. So yeah, I'm going to talk about a bit more practically about using the Polygenic Score Catalog Calculator, which is a reproducible workflow for PGS calculation. So what is it? So if you have some imputed human genotypes, which are called target genomes in sort of in PGS, if you can open a terminal and run some commands, because it is a command line application, and you want to calculate some polygenic scores, and you want you want to make things that look like these figures on the right, and you want to contextualize your calculated PGS with a genetic ancestry analysis, then the PGS Catalog Calculator will probably be helpful for you. So why is the PGS Catalog Calculator different from other software? Because there's lots of software that will calculate polygenic scores. So the calculator is very portable. So a lot of genetic information is unable to leave a particular computer for data privacy reasons. But the calculator has been deployed to offline environments. It's been deployed to trusted research environments, places like UK Biobank, places like FinGen, things like that. The calculator has very deep PGS catalog integration, so you can calculate when you're working with the calculator, if there's a specific publication or a specific trait or a specific PGS ID in the catalog you want to work with, you just describe the accession that you want to work with, and the calculator will sort of take care of grabbing that information from the PGS Catalog API and downloading all the data in the correct genome build in a nice format. The calculator will automatically process the data so that you're able to calculate hundreds of scores in parallel if you want to, if you've got a big enough computer. So it's much more efficient to actually calculate. Sort of if you had, for example, a cohort of say 1,000 people, it's way more efficient to run the calculator once on your cohort rather than running it once for each score or even once for each individual. That would be sort of the least efficient way of doing things. So the calculator also includes a robust genetic similarity analysis. And as I mentioned before, it's been widely deployed to trusted research environments. There's all of the software that we make that's sort of included in the calculator is quite thoroughly tested. There's lots of documentation. We also have a GitHub repository with a discussion forum. So if you have any problems, you can create issues or create discussions. And there's a really good community support available as well. If you wanted to theoretically follow along today, you would need to be working on Linux or Mac OS with an open terminal. You would need Nextflow installed and Docker Desktop. The calculator does run using other methods, not just Docker Desktop, but for ease of demonstration, I'll just be using Docker. And also, if you do install Docker on your computer, you need, like if you've got a work laptop, you would need permission to install that software. So the way you install the calculator is by running the test profile. And what the test profile actually does is it fetches our code from GitHub. It copies it to your computer. It downloads things called Docker containers, and what that does is it contains all of the software that we create and also some software that we reuse, like Plink. It fetches that and puts it onto your computer. So the first time you actually run this, so on the left here, this video is just showing what that looks like. And the first time you ever do this, it actually takes quite a bit longer than this video because the containers are roughly about 2 gigabytes. So depending on the quality of your internet connection, it can take a little bit longer. And what the test profile is doing is it's analyzing a very small, tiny synthetic data set. So the results don't actually mean anything, but what's happening is all of the steps that you need to happen to calculate polygenic scores are then running on your computer. And the reason why this is really helpful is because if you can run the test profile and then you encounter successfully, and then when you're trying to apply this to your own data, if you then encounter problems, you can be sure that your computer is okay and that the calculator is okay. It's probably then much more likely to be a problem with your data or with some sort of configuration that you've set up. So this is the first step. Running the test profile takes care of actually installing the calculator on your computer. So what you get out of the calculator, one of the main things is this report. So it's a HTML report that opens up in a web browser in your results folder, and it includes metadata about the workflow. So it describes it's like a log of the things that you've asked the calculator to do. So it's reproducible. So it describes things like the scoring file, metadata about the scoring files, about the version of the calculator. It will grab information from the PGS catalog to describe like the publication, the number of variants in the scoring files that you've used, and things like that. The next biggest section is about variant matching, which I'll describe a little bit more in the next slide. But this is probably the most important step in the calculator. So when you have a scoring file in the PGS catalog, there are probably thousands of rows, and each row describes a different genetic variant. Many of the biggest scores contain millions of variants. And there's no guarantee that the variants that the authors of the scores have used to create the polygenic score actually exist or are described in your data. So the main aim of the calculator is to faithfully reproduce these published scores in new target genomes. So if you are running a score or calculating a score, and you find that actually there's very few of the variants in the scoring file are actually present in your data. So for example, if there's a score with 10 million variants, but in your data there's only say 100,000 or a million or something, you're not really actually calculating that polygenic score. You're missing lots and lots of data. So this report is very important to have a look at. To make sure as a sort of a quality control check to make sure that what you're doing is sensible. And there's sort of more detailed statistics about the variant matching process. So in this example, in the test profile, we can see that like 100% of the variants on one score matched, and then like 93% of the variants matched in another one, which is quite good. And by default, the calculator will have an error message, or it will refuse to calculate scores if fewer than 75% of the variants in all of the scores that you request are present, just because you're not really actually reproducing the published score. And that is user configurable. So if you still want to go ahead and calculate a score, you can change that, which is described in documentation. So this is the actual scoring file that's created by the calculator. So it's a very simple tab delimited text file that's compressed. So for each row here is a different sample in the cohort, and then each column is each row is a different PGS with a different value. So this is the standard output from the test profile. Another output here is the matching log. So this is very important when you're first getting started, is to make sure that the variants. This is how you can check to see. So if you're really interested to know why a variant isn't matching from the scoring file, if it's not found in your data, this will describe why. This will describe how the things we've tried to make sure that that variant is present in the data. So we try lots and lots of different things to try and make sure that we can match that variant. And this log describes all of the different candidates that we had that were generated. So it's very useful for auditing and debugging and understanding of what went into the score. So that's quite helpful. So as a quick reminder, as Sam mentioned, ancestry adjustment, contextualizing polygenic scores with genetic ancestry similarity analysis is really important. We do describe in a lot more detail in our documentation on the bottom here. And there's lots of optional components in the calculator that sort of get activated. So the test profile didn't actually include any genetic ancestry similarity analysis tool. It just ran a very simple calculation process. But these bits that I've highlighted here are the bits where the reference genomes, the reference panel, are actually used and incorporated into the matching and scoring processes. And as a reminder, again, there's these two sort of broadly speaking, there's these two types of normalization methods, one of which is empirical and one of which is PCA based. So if you're then looking to use the calculator with your own data, the first thing you need to do is set up a sample sheet. So a sample sheet is basically a spreadsheet that you can create using Excel or something, or any spreadsheet software that describes your data. So your data should be a single human cohort. It might be split by chromosome, that's okay. In terms of file format, so it should be imputed as well. So typically a lot of PGS are actually developed using imputed cohorts. So then it's very important if you've got when you've got array-based data to make sure that your data has been imputed as well. And it might be split by chromosome. And in terms of file formats, we support plink1 files, plink2 files, and also VCFs. And it's if you do split your data by chromosome, it's really important to include all of the chromosomes. Lots of people try and run one chromosome as a test, and then they get an error message saying that they had the variants weren't matching very well, and it's actually matching fine. All of the variants on chromosome 1 were matching, but the variants on chromosomes 2 to 22 just weren't matching because they weren't there. So it's really important to include all of them. So once you've got your spreadsheet ready, which describes the name of your cohort, the paths of the files, which chromosome each file is associated with, and the format of each file, you're then ready to run the calculator. Possibly this example here uses ancestry adjustment. So on this example, the target genomes are the Human Genome Diversity Project, and all of this data is publicly available. You can download this from the Plink website. And the reference panel that we're going to use is, on the example, is 1,000 genomes, and it's calculating PGS 001229, which is height, and we're including the genetic ancestry adjustment. So it's really similar. So the only difference really here is, so all of the commands are very similar. It's one extra parameter basically, and then sort of the workflow will take care of the rest. But you'll see, so the biggest changes here are the input is the sample sheet. So the spreadsheet that you've created in Excel has to be in this format here, the CSV format. And then you've got to specify the target build of your genotypes. So we support 37 to 38. Then the ID of the different polygenic scores that you want to work with. On this example, I've included the cache because it's this would take quite a while to run in a live demonstration. So this is cheating a bit. It's going to be really fast. And then I'll pause here quickly. So this run ancestry parameter, which points to the path of the reference panel. We provide 2 reference panels on our website. One is 1,000 genomes, and one is 1,000 genomes combined with HGDP to include sort of greater diversity of populations. And then everything else is quite similar. I've removed the test profile because we're no longer testing. And what you'll see is that when the next flow pipeline starts, it's going to run very many more processes. So you can see all of these processes are all cached. So it's a lot more work it's going to try and do. It's a lot more things happening. But that's it running the ancestry adjustment. So in reality, this would take much longer, probably depending on the size of your data set, maybe a couple of hours. But yes, for this demo, that's what it looks like. And the enhanced output. So what changes is so you still get the same report. What changes is the output. So you get some additional columns. So you get the empirically adjusted score columns here. So the most similar pop and the percentile most similar pop. And in the report, you also start getting these plots here with genetic ancestry plots and population similarity summaries as well. So you also get the PCA adjusted columns as well. So really, that's the only difference in terms of the outputs is these extra columns in the scoring file and extra sections in the output report and the HTML. So then, very briefly, what can you do with the PGS output? So people use PGS to model genetic predisposition to different exposures or outcomes, and they also they might use the principal components that the calculator also produces, or population similarity labels to assess how well PGS transfers to different populations. But there's lots of things you can do with PGS, which is sort of out of the scope of this webinar. So in summary, the PGS Catalog Calculator can portably calculate PGS at biobank scale. It supports automatic genetic ancestry estimation and will adjust calculated PGS in the context of genetic ancestry. We do have a paper out in Nature Genetics if you're interested to learn more, there's a link in the top right. And then depending if you're interested in the PGS Catalog or PGS Catalog Calculator, there's links that are available here as well. So for the calculator, the main place to go is to our GitHub repository, which is where all of the links that you need to work with the calculator are. And that's where you can ask questions or get help if you're unsure about anything. But yeah, I think now there's time for questions, I think, which we will go through, I guess. So
thank you very much, Sam and Ben, for today's webinar. And thanks everyone for joining in. So I think I can read out the questions and then you can take it. Is that fine with you?
Yeah, that sounds good.
All right. So we can start from the top. How can we set the range of low risk, high risk, and normal in PGS Calculator?
So I think basically what happens is that we'll give you back the relative. If you calculate the polygenic score with the catalog and use the ancestry information, this is going to give you the PGS for that individual person, and then how many standard deviations higher or lower than the mean for people of their genetic ancestry does this person have. So in terms of setting what's lower, higher, and normal, I think here, I guess you have the in reference to the mean, so that provides you the orientation of whether someone's higher or lower. But in terms of setting the threshold at which is someone is high or low risk, this comes down to you who's using the data. So setting a low or high risk might be more about the effect size that threshold confers on the phenotype that you're testing on. But there's nothing inherent in the PGS distribution that we can use to set low or high risk. We can also just provide where someone sits in a population. And then if you said you made a threshold of like 2 standard deviations above the mean and found that those people have like 5 times higher risk and you deem that higher risk, that's mostly up to the end user.
Yeah, thanks for the answer. The next question is, how about sample overlap, discovery versus application sample, and how is that handled?
Right. So for a polygenic score to be included in the polygenic score catalog, you basically have to show that the polygenic score has predictive validity in external samples. And so the data sets do have to be overlapping. They have to be non-overlapping, or at least some sort of cross-validation to show that it's not overfit. So in terms of sample overlap, we don't handle anything like that in the catalog. That's up to the authors when they're developing the score. But what we do show is which samples were used to train and test the score. And so if you know that the score has been evaluated and used in this cohort, and it's labeled as such in the PGS catalog, then you know it's probably safe to use in your own applications. So basically, if you're curious about sample overlap, you should look in the metadata of scores in the catalog and then just look at which cohorts have been tagged for the development and evaluation samples, and use that to guide which scores you use in your research.
Great, thank you. So the next question is about the terminology, and I think I would have also given thumbs up to this question. Regarding terminology, a PGS PRS is a score that is a single number associated to an individual. I wonder if it would be more clarifying to refer to the term PGS model. Some studies use this term as the aggregation of the development samples plus statistical method, which equals scoring file. This is usually referred to as score or PRS, but gives rise to confusion in conceptualization. What is your opinion on that? Would you say that PGS catalog is a database of PGS models?
I think Diana's right. I think the PGS catalog is a catalog of PGS scoring files, and then the metadata about how those scoring files are useful and how well they predict the trait of interest. I agree that the term PGS or the score is often more particular to the person, like at individual level. And indeed, like the Polygenic Score Catalog Calculator calculates individual polygenic scores for the person. So it is fair to think of the catalog more as a catalog, it's not a catalog of individual level scores. Data. It's a catalog of scoring files that you can use to calculate individual level data in different cohorts. Ben, do you think I explained that?
Yes. Yeah.
Okay.
Thank you. To validate PRS model using a specific population, how big should my cohort size be? If the PRS model is previously validated using subset of UK Biobank data.
I think this is a hard question to answer because, obviously bigger is better because that increases your power and the ability to detect an effect size. I think how big the evaluation cohort you need is probably more proportional to the effect size you think your PGS is, so if you look at, or if you're thresholding the PGS, it's about the prevalence of being high risk and what the effect size is. So if you're interested in trying to figure out how big a cohort should be, this would be sort of traditional power analysis, similar to what you would do for a GWAS or any epidemiology study, where you need to know something about the prevalence of your risk factor and the effect size, and then it will sort of tell you in the range of the sample size of your cohort that you would need to evaluate it or have proper power to detect that PGS association.
Okay, thank you. Next one is, do I understand correctly that PGS catalog applies, sorry, PGC catalog applies the same method for PGS calculation on your genetic data as was used in source paper? For example, LD-PRED2 is applied in the source, then the same will be applied on my samples to calculate PGS.
Yeah, so I guess there is a distinguishing. So LD-PRED2 is a method where you give it some GWAS summary statistics and individual level data, and it will do two things. The first thing it will do is figure out which variants and weights that you want to include in the polygenic scoring file that's then used to calculate the polygenic score on individual level data. And so it does using a statistical method like a Bayesian framework. And so, but then once you have that scoring file, the variants and weights, and then apply it to those people, it does that using the standard weighted sum of effect sizes times dosages aggregated across all the variants per individual. And so it's that second step that once you've chosen the variants and weights to calculate the polygenic score with on a person, the PGS Catalog Calculator uses the same aggregation procedure as LD-PRED2, which is fairly standard across all PGS software. So the PGS, our tool doesn't do any development in terms of selecting which variants and weights should be in the polygenic score, but once you have a fixed polygenic scoring file, the variants and weights, it will do the aggregation the same as almost every other tool.
Okay, thank you. Next question is what about population with diverse ancestry to generate genetic ancestry supported PGS score?
So I think this refers to what's the right reference panel or groups of people used to compare to. So basically, it's important when you're calculating the relative polygenic score to compare that person to a diverse set of people or a set of people who are representative of their ancestry. And we give two versions of the reference panel, as Ben said, for comparison. There's one that uses 1,000 genomes. That's the 5 main superpopulations from the original 1,000 genomes paper. And a second paper that a second reference panel that's 1,000 genomes plus data from the Human Genome Diversity Project, which then adds additional superpopulations for comparison, so Oceanian and Middle Eastern mainly. And so it's important that when you calculate a polygenic score and that you compare it to the right people so that you can get the most accurate representation of the relative genetic predisposition of this person. And so we do that by providing sort of the most diverse reference panel that we can openly. But you might, if you're comparing within a cohort, maybe you have a specific reference panel already that you've used for imputation and things like that. And so that might be more appropriate in these situations.
Okay, thank you. Can we calculate the score with a target genome that consists of SNP data from the same chromosome, but from various individuals in the same file?
Yeah, so that's just a cohort. Yeah, absolutely. So if you have multiple samples in your target genotypes, that's totally fine. That's encouraged, if anything. So as I think I mentioned, if you have your target genome split by sample, it's much more efficient to merge them before you put them into the PGS Catalog Calculator. And as a reminder, there were three types of format that we support in the calculator. One is VCF, and the others are the plink 1 and plink 2 formats. So they really do work best with cohorts. And we encourage people to always use cohorts with the calculator.
Yeah. And I think something we're working on in our future software development plans are to make the Polygenic Score Catalog Calculator work better for individual level data, like individual samples, and data from different whole genome sequencing formats is something on our roadmap.
Right, thank you. So the next question is, I think, is looking into the bigger picture of the application of PGS Calculator. What are the applications of this research in our daily life?
I think that that's definitely an interesting question, and one that definitely should be asked is what is the point of this research, and does it contribute to health and understanding? And I think polygenic scores are a tool basically to predict predisposition, but they might also be clinically useful. I think there's applications where if we knew who was at genetic risk for a disease, maybe we could target different screening regimens, like more advanced screening regimens or targeted prevention at individuals. And so that's sort of the idea of polygenic scores, like calculating these genetic predispositions might be useful for healthcare and also for researching, trying to identify pre-symptomatic people who are likely to develop a disease, these types of things. On our side, I guess in this project, we're basically providing the data so that people can go out and do those experiments. And we're also providing the software to make the polygenic score calculation most reproducible and easy to use for end users. So I think the daily life is that we make this software, this database and these software tools easier for scientists to go out to do the research that might then impact. This is enabling work to help people make the impact, other scientists to make impact as well.
Great, thank you. Yeah. So when running PGS Calculator, does the summary statistics have to be in the PGS catalog? If not, could we choose what PRS method we want to use from our GWAS summary statistics, LD pred, precise, etc.?
So you would have needed to run those other algorithms like LDPED or precise before using the PGS Catalog Calculator. But the outputs of those will be compatible with use of the PGS Catalog Calculator. It doesn't need to be a score that's existing in the catalog. As long as you give a score with the variance and the weights and the alleles, it will be able to calculate that. So basically, the score the calculator basically works with data that's user supplied as well.
Now, this next question refers to an earlier question, which was about low risk, setting up low risk, high risk. If the phenotype for each individual is known, could one create two distribution plots, one for cases and one for controls, and then determine the high risk threshold by identifying the value that best separates the two distributions?
Yeah, I think that's definitely one way to do it. And that's an example of an application of how you would use the results from the catalog calculator, combine them with phenotypes to do other interesting research.
To predict PGS score, if cohort size is just 1,000, how would you say true representative of a large diversified population metadata to predict PGS?
I think that's a good, like, how do you know that a polygenic score works in the samples, or that that sample that it was evaluated in is representative of a larger population? I think that's just the same for any risk factor association you see in the literature. It's up to you to sort of look at the sample size, how that sample or cohort was created, the effect size, what other variables were included in this model, and judge and take that information and holistically interpret it according to sort of the best practices. So that's why we provide all the information in the catalog of the metadata on who the samples were, who the score was evaluated and developed on, and so that you can make those judgments and interpret whether you think that the effect size they measured is likely to be seen in a new cohort of people of similar characteristics.
Thank you. So we have about 8 minutes left, but we have questions coming in still. So I'll take the next one. How would you suggest to model the effect of a particular exposure on the predictive ability of a PGS for a trait?
You could use the results of the calculator for the PGS, but then I think I would look into the literature about gene environment interaction studies that use polygenic scores as an exposure of genetic predisposition. So I think there is a building literature about G by E. I think there's actually a course at EBI coming up next February or March about G by E, where we'll talk about polygenic scores as well. But it's definitely an area of research, and I would suggest to look up G by E studies, gene environment studies that include polygenic scores as an exposure.
Great, thank you. Yeah, we also have a collection based on this course, which was published earlier this month. I'll share the link very soon in the chat box. Okay, so next one is, what criteria should be used to select the most suitable PGS for a specific trait, given that the catalog contains multiple PGS options for the same trait?
I think that's a good question. I think I would look at a couple of things. I would look at what was the sample size and diversity of the GWAS in development cohorts. I think oftentimes polygenic scores that were trained in larger subsets of people are more accurate and usually contain more variance and better estimated variant effects so that it could be used. And I would look at how that polygenic score had been evaluated and see in which people, what effect sizes, was this consistent across cohorts of individuals with the same ancestry? Is it consistent across different ancestries? So I'd look at all those pieces of information and then try and find the score that seemed to perform best in the situation, the scenario that's most similar to the one I wish to apply it in as well.
Is there any additional filtering required for the VCF file, such as filtering based on allele frequency or other criteria before using them as target genomes in PGS Calculator?
Sure. So that would, I guess, that would influence the matching process. So I don't think you have to. That should be totally fine to just use the unfiltered data. If you do filter those variants out of your data, that will mean that fewer of your variants will be able to match against the scoring file. But yeah, I guess if you have concerns about the quality of those variants, you might want to do that and accept a lower match rate. What do you think, Sam?
Yeah, I think that's definitely true. It's like if you think the variants aren't genotyped correctly, it's probably best to exclude them from the PGS, the input to the calculator. In the calculator, we also allow you to prune or to include or exclude variants that are multi-allelic or that are ambiguous, so like palindromic, because these might cause matching problems. All of this is customizable, but basically, we don't do too much QC within the catalog calculator tool. We mostly leave that to you to do before, because just no variants that are incorrectly genotyped should enter in. That's somewhat the assumption there.
Right. What are the choice of scale, additive or multiplicative, to obtain the interaction between traits?
Again, I think that's not really within the scope of our talk or the tool. I think you should how to evaluate gene environment interactions and is an open area of research, and there's a lot of methodology out there. So I would encourage them to read some reviews on the topic. Yeah.
Okay. How does a polygenic risk score, which is based on the linear addition of effective sizes, compare to methods that explore nonlinear associations between genetic variants? For instance, how would a machine learning-based model for calculating risk scores perform relative to a traditional PGS approach?
That's a good question. I think, so I should say that right now we have it doesn't only calculate additive scores, it can also calculate scores with variants that have recessive or dominant genotype encodings. So that allows basically some non-additivity to be captured within the calculator as such right now. I think how these traditional polygenic scores compared to machine learning based methods is an active area of research. papers I've been involved in have sort of seen modest improvements with machine learning based methods. I think you get better performance from just better GWASs and better diversity of samples, less so from fancier methods. The fancier methods and more advanced methods can improve some things, but I think heritability estimates suggest that most genetic variation that leads is additive in nature.
All right, thank you. So we just have 1 minute left, so I can take this last question. Is there a method to normalize PGS values when analyzing associations between comorbidities, particularly if planning to use a PCA-based or multiple PGS scores from multiple traits?
I'm not sure. That sounds like a really interesting research topic, one that's close to my own research heart. But I think it will depend on your situation and what sort of associations and clustering analyses you're doing. So, but I think you can use the results of the calculator as input to those types of analyses, definitely.
Great, thank you. I think it's just one more question, so I think we can we can take that.
Yeah.
Would it be difficult to calculate PGS for rare disease based on Ancestry or GWAS database?
I think so. Scores for rare disease are hard because they're caused by very few variants, and the exposure is rare. The PGS as well as the outcome, so it becomes hard to predict. So polygenic scores aren't the best tool for that. There is a lot of development in the literature about how you incorporate rare variants into existing polygenic scores or model polygenic scores alongside rare variation that causes somewhat common or rare disease. I think that's something that the PGS catalog is looking at about how to support polygenic scores that include these rare variants and make sure that they're usable and accessible to researchers. And so I think stay tuned to our Twitter, our website to see our developments on that.
Great. So that concludes our webinar today. So thanks again, Sam and Ben, for delivering the webinar today and answering all the questions. And thanks everyone for joining today and yeah, asking lots of questions. All right. So with that, thank you very much.